Hello, ladies and gentlemen. This is Amrita Ramesh, and I am here with a video from Cell Structure and Function. This is from class 11 NEET Biology. Here in this video, I'll be discussing about prokaryotic cell. Before we move on, I would like to notify that all these video lectures are available in Tamil as well. You can kindly share it with your friends and relatives who would for an explanation in Tamil. So this slide is about my background, all introduction about myself. So let's move ahead topic. In the previous spoke about cell theory, cell life, what are the types, types, and all of that. So let's move ahead. Prokaryotic. In this, we'll detailed study about the general characteristics. So before we move on, quick recap. Let's finish cell. What is a prokaryotic cell? A cell that does not have a true nucleus. I think everybody is quite familiar at this what true nucleus means. True nucleus is when inside a cell there is a nucleus which is covered by a nuclear membrane. If that kind of a structure is present, we call that as a true nucleus. But here, in case of prokaryotic cell, the cell does not have a true nucleus. It is the genetic material or the DNA that is lying naked in the cytoplasm. So that is exactly what a prokaryotic cell is. So the example of a prokaryotic cell is a bacteria. Now, what are the other unique features of a prokaryotic? There are no cell organelles. What do you mean by an organelle? Something that has its own function, membrane-bound structure with a specialized function is what is called as an organelle. Now, here we do not have organelles, no cell organelles present in prokaryotic cell. There's no true nucleus, there's no cell organelle, and that is therefore there is no division of labor. So, what does it mean? One single cell performs all the functions. Let's move ahead and look at the characteristics of a prokaryotic. Now let's take example of a bacterial cell here, and further in the slides we'll again study a detailed characteristic study of a bacterial cell. What exactly is a bacterial cell made up of? What are the other inclusions that are present in bacterial cell? But first, let's have a quick look at the prokaryotic cell characteristics. So the first point is nuclear material lies naked in the cytoplasm, which is called as a nucleoid. So when the genetic material, the DNA, is lying naked in the cytoplasm, we call that as a nucleoid. No true nucleus covered by a nuclear membrane. So this is the most characteristic feature of a prokaryote. They are all primitive organisms. Cell wall is present. And in this also we have an exception, which is a mycoplasma. Mycoplasma is the smallest bacteria. Cell wall is present. So when I talk about cell wall, three organisms have cell wall. One is plant, another one is fungi, and the third one is bacteria. Bacteria also has a cell wall. So cell wall is present. There is no true nucleus. They have external structures. What are the external structures? Flagella, fimbriae, and pili. So flagella, I think most of us are familiar with. Flagella is just like Euglena has a flagella. If you've seen Euglena, Euglena has a flagella and the function is motility, okay, for propagation, for movement. Similarly, in this case also, for bacteria, they have flagella. And this flagella also serves the same purpose. It also helps in motility. The flagella helps the bacteria in moving forward or backward. Either ways, the flagella helps in movement. Next, so we have two different structures. One is fimbriae and one is pili. Fimbriae helps in attachment. Now, supposing a bacteria is swimming in a pool of water and it finds a substratum. What is a substratum? A rock or something where it can go and sit. Now, if there was nothing to attach itself to, how will it sit? It will just slip off. So, these fimbriae are those structures which help in attachment to a particular surface or a substratum. So, in this diagram, if you see these small structures, they look exactly like cilia. So, paramecium, if you've seen paramecium, they have cilia like this. But the function of cilia is also motility. Fimbriae do not help in motility, they just help in attachment to a surface. So that is the function of a fimbriae. Now, what is the function of pili? Pili, if you see this, they are slightly longer in length when compared to fimbriae. They also have attachment purpose. They can also serve the purpose of attachment. If the bacteria wants to go and attach itself to a particular substratum or a rock or anything, pili helps in that as well. And one important function it has, which is that of your mating. So, I'll explain that in a little bit detail. Now, supposing you have this bacteria, this is one bacteria which has a particular DNA material. Now, supposing this bacteria wants to donate it to another bacteria which is adjacent, which is present here. Now, it takes help of this pili. So, pili functions as a bridge. That is the reason we have given the name helps in conjugation. Okay. So via this tube, so this pili helps in forming this tube-like structure. Via this tube, the DNA material enters this particular bacterial cell. So this is one bacterial cell. This is another bacterial cell. Now this bacterial cell wants to donate its DNA material into this. Okay. Now how will it do it? So with the help of this pili. So pili forms a tube-like structure and it donates this DNA. So the DNA will cross this tube and then go enter into this. So that is function of pili. Next, photosynthetic thylakoids are present. So as soon as we hear the word thylakoid, the first thing that comes to our mind is chloroplast. So always we associate it with chloroplast in plants. So here also the same function thylakoids have, but they are present in bacteria, and we call these bacteria as photosynthetic bacteria. They have the ability to photosynthesize, and we have a specific name for that as well. They are called as photoautotrophs. Photo means light, autotroph means they can prepare their own food. Photoautotroph. So, photosynthetic thylakoids are present in photosynthetic bacteria. Next, cytoplasmic inclusions and gas vacuoles are present. Now, what is the function of gas vacuoles? Gas vacuoles helps in buoyancy. Supposing we have so many bacteria that are living in the sea. Now, they can't go deep inside, right? It is very difficult for them to get oxygen or anything. Okay, if supposing there is a photosynthetic bacteria, if a photosynthetic bacteria lives deep under the sea, it will not be able to get that much amount of sunlight for preparing food. So that is the reason they have to be on the surface. Now, how will something come up on the surface? It has to develop this tendency of buoyancy. So these gas vacuoles will provide buoyancy. They provide that buoyant force for it to propel upwards and stay on the surface. So that is the function of these gas vacuoles. The cytoplasmic inclusions are nothing but storage organs. They have PHB granules which help in deriving energy for the bacteria. So they are basically nutrient storage uh, vacuoles or nutrient storage bubbles. Okay. Next, ribosomes of prokaryotes have a sedimentation coefficient of 70s composed of 50s and 30s. Now, this is a very important point. There is a possibility that there can be a question. So, ribosomes of prokaryotes and ribosomes of eukaryotes. Okay, ribosomes of prokaryotes are 70s, whereas ribosomes of eukaryotes are 80s, and they are composed of 40 and 60, or I would say 60 because that's a bigger unit. 40 is the smaller unit, smaller subunit. So here it is 50 plus 30. Remember one thing very clearly here, never try to add 50 plus 30 or 60 plus 40 because that is not going to get you 70 or 80. It is not based on the addition, it is based on the sedimentation rate. Now, what is the sedimentation rate? If you have seen our washing machine or a mixer, okay, what happens? There is a centrifugal force there. Okay, so when something spins, okay, the denser particle will be at the bottom and the lighter particle will be at the top. So that is the concept of centrifugation or centrifugal force. We are trying to separate things based on the density. The principle is density. Similarly, here also ribosomes have a particular density. Nucleus has a particular density. Okay, mitochondria has a particular density. Supposing I have a cell and I want to separate out all the organelles. Okay, I want to study a single organelle, what mitochondria does, what chloroplast does. I want to study it more. Now, how will I separate it? Because they have a particular density. So I will spin it. I will put it in a centrifuge, spin it at that particular RPM. Okay. At one particular RPM where I know they will get settled out. Okay. Either at the bottom or at the top, depending on their density. Similarly, ribosomes of prokaryotes also have a sedimentation coefficient. Now, if you spin it together as such, they settle at 70 S. S is Swedberg unit. It's a unit to denote it. To denote the sedimentation coefficient, we have this unit called as S. S is nothing but Swedberg unit. Now, if I am taking the prokaryotic ribosome just like that and spinning it, I am observing that it settles down at 70, at the position 70. Whereas when I'm trying to separate the two units, okay, ribosome is basically made up of two units. I'll draw it here for you. So this is the bigger subunit and this is the smaller subunit. So in case of prokaryotes, it will be 50s here and 30s at the bottom. And in case of eukaryotes, it's 60s at the top, that is a bigger subunit, and 40s the smaller subunit. So this is how ribosome looks. Now together they will settle at 70. But if I'm separating it out into 50 and 30, now I don't know if it's 50 and 30. I know there is a bigger subunit and a smaller subunit. Now if I've separated it out and I'm seeing where exactly it's settling, the bigger subunit will settle at 50 and the smaller subunit at 30. So this is why we have 50s and 30s. Similarly, it goes for eukaryotic cell as well. So don't try to add it up ever. 50 plus 30 or 60 plus 40 is not to be done. Next, location of both transcription and translation is in the cytoplasm. Now that's very obvious, right? Because a prokaryotic cell does not have any organelles, does not have any compartmentalization at all. There is only one single cell, there is only a little bit of cytoplasm, and the nuclear material is just lying there. Okay. Now, if there is no compartmentalization, everything will happen at the same place, unlike a eukaryotic cell. Okay, there are no division, there are no rooms, there is nothing. Okay, everything is one single cell. So all the activities will be taken uh, care of by that one particular cell, and it will happen in the cytoplasm only. But in case of eukaryotes, there is a nucleus, there is a nuclear membrane. Okay, now transcription will happen inside the nuclear membrane, but translation will happen in the cytoplasm. So for that, the mRNA has to come out. Okay, come out of that nucleus. Nucleus is one room, cytoplasm is another room. So from one room it has to come out. But in case of prokaryotic cell, all that is not required. We do not have to do that because only one single cell is present. Only one single cell, little bit of cytoplasm and the nuclear material. So everything will happen in the cytoplasm. Again, a very important question. In some bacteria, spores or cysts are formed. Okay, so spores and cysts have a similar function, just like fungal spores and cysts. So fungal spores, what do? They are formed when the conditions are unfavorable. Now, what do I mean by unfavorable condition? When the weather is not correct, when the temperature is not correct, when the humidity or pH, okay, or nutrient availability, all that is not favorable. If it's not correct, the fungi will not resort to growing. It will instead become spore and become dormant. Okay. Now we can take examples of bears. When the temperature is very, very cold, okay, they go and hibernate, right? They just go and sit because they want to conserve energy. Similar concept is used here also by the bacteria or fungi. Okay. They will resort to forming spores to tide over that particular weather condition. Okay, once the weather is fine, okay, once the availability of nutrient is fine, sunlight, everything comes back to normal, they will germinate. They will germinate and start doing their activities. Okay, because doing an activity requires energy, and when the condition is not favorable, it is not advisable to spend that much amount of energy. So that is why some bacteria, they will form spores and cysts. Another very important thing is a plasmid DNA. Plasmid DNA is a circular DNA which is present inside bacterial cell. This is used in recombinant DNA technology. So when you start studying about recombinant DNA technology in the next few chapters or in the 12th standard chapters, you will understand the importance of plasmid DNA in a better way. Why is it so significant? Why is it used for? Because it has a huge role in recombinant DNA technology. So these are all the points for general characteristics of a prokaryotic cell. What do they have and how exactly it is present. Next, we'll move ahead to see a bacterial cell, specifically when we start talking about a bacteria. So first of all, what is the average size of a bacterial cell? It is about 3 micrometer to 5 micrometer. Now 3 micrometer is 0.003 centimeter. Okay, if you're not understanding in terms of microns, it is about 0.003 centimeter. Okay, it is very tiny, extremely tiny to be viewed through a naked eye. So we need to have a special instrument, that is a microscope, to observe the bacterial cell. Now there are first we'll study about the different shapes and types that are found in nature. Okay, what are the different types of bacteria that are present in the nature? What are the different types and what are the different shapes of bacteria that we observe? The first shape is a coccus. Now what is a coccus? Coccus, the word means spherical. Okay, it means a sphere. You know, sphere, right? Three-dimensional, when we say it's a sphere, and two-dimensional it's circle. Okay, three-dimensional sphere. So this is what is a coccus. Now, if this coccus is present singly, okay, if I'm doing a bacterial cell culture and I'm observing it under a microscope and I observe these single structures, only one circle everywhere throughout the slide, I will say it is a monococcus, okay. Mono means single. So it's a monococcus. Now, if I see something like this, if I see something like this in pairs, okay, if I see something like this in pairs, I will say it's diplococcus. So two single coccus are joining together. They are occurring in pairs. So that is when we call it as a diplococcus in groups of two. Now, if you see something like a chain, okay, a chain of coccus, we call it a strep. Always remember, strep means chain. So if this coccus is seen in chain as a chain, we call it as a streptococcus. Next, if you see as random clusters or random bunch, okay, just like you see have a grape bunch. Similarly, if you see it under the microscope, some random bunches of coccus, you say that is a staphylococcus. Next, if you see in groups of 4 or 8, okay, multiples of 4, then it is called Sarcina. The name is Sarcina. So in under coccus, you have so many different arrangements. Coccus is a particular shape, and these are all the arrangements possible. Okay, these are all the arrangements possible under coccus. Next, we'll come under bacillus. Bacillus is a rod-shaped or cylindrical bacteria. Now, if you see here, this is how a bacilli is. They're elongated, cylindrical, rod-shaped, elongated bacteria. Now again the same thing applies here also. If it's a single, we just say it's a bacillus. Bacillus is singular, bacilli is plural. So occurring singly, we say it's a bacillus or bacilli if there are many. If they are paired, okay, if they occur in groups of 2, you use the same prefix, that is diplobacilli. It's just diplococcus, here it's diplobacilli. So the same prefix will be attached here as well. Similarly, if you see chains of bacilli, we call it streptobacilli. Then, if they are stacked on one top of the other, then it is palisade bacilli or bacillus. Example here is a lactobacillus, that is milk. Milk, the bacteria found in milk is lactobacillus. Now, here the example is Staphylococcus, Streptococcus, all of them are example. That is why I haven't specifically written any other example. So here the example is Strepto, Staphylo, all of them, Sarcina, all of them are examples themselves. Next we'll move on to the next type of or shape that is observed. So next we have spirillum. Spirillum is nothing but a screw shaped corkscrew. Now if you've seen a screw, we all would have seen a screw. Screw is something like this, right? Similar is this shape here. If you see here in this slide, we have the corkscrew shape. So it looks like a screw. It's screwed. Okay. That is what is a spirillum. That is again a different shape of bacteria that is found. And here no arrangement is observed. Just like we said in coccus, there are different arrangements possible. Monococcus, diplococcus. Okay. Here in this case, in spirillum, there is no arrangement. They do not get arranged. Okay. They do not get arranged into any forms or groups. They occur singly. Next, we have another very common shape is a vibrio. Vibrio is a comma shaped. Now if you look at here, this diagram is a vibrio. Okay, they exactly look like this with a flagella on one end. Okay, so they are comma shaped and they are curved. Here also again they occur singly. There is no aggregation or no grouping that happens in case of vibrio as well. Now example is Vibrio cholerae. Vibrio cholerae causes cholera. Okay, Vibrio cholerae causes cholera. Some other shapes, which these shapes do not occur very commonly. Okay, they occur a little rare. They do not. We are not very familiar with them because we do not find them in our normal daily lives. They are present in very rare conditions. Now, I would like to tell about this because here we can see some spores. So I want you to see how the spores look. This is a bacilli. You can see it's a elongated bacteria. Okay, it occurs in chains. Okay, and there is a spore. In the center, you can see that part hasn't got stained. The whole organism gets stained, but this part has not got stained. So that is nothing but a spore. Okay, and this is nothing but anthrax. This is the anthrax bacilli. So Bacillus anthracis. This is Bacillus anthracis. Now, what is so different about this bacilli is it is rods with squared ends. Okay, if you see this rod, it is cut right on the edges. It's cut, so it is squared. Unlike this bacilli, which is curved, it has a curved ending. But this anthrax bacilli does not have a curved ending but squared ends. Okay, so that is what is a feature of this anthrax bacillus. And this one is nothing but a lobed spears. Okay, so it has two lobes if you see. So let's move on to the next topic, that is flagellation. Now we spoke about the function of flagella. What does the flagella do? It helps in motility or movement of the bacteria, either forward or backward. Now based on the flagella or position of the flagella, again bacteria is classified. Okay. Now the first important classification is whether they have flagella or they don't have flagella. If a particular bacteria does not have a flagella, we call it as atrichous. Atrichous. Now trichus means flagella. A means no flagella. So there is no flagella. Atrichous means no flagella. Next, if it has a single flagella, it is called again mono. Okay, mono means single. So monotrichous, a single flagellum present at one end of the bacterium. Similarly, so like cholera. Cholera bacteria is monotrichous because it has one single flagellum. When you call it as flagellum, it is singular. So it has one single flagellum present on one end. Now amphitrichous, what is amphitrichous? On both the ends. Now we know amphibian or amphipathic, both ends or both. Okay, when we use the word both, we use the word amphi. Okay, we know the plasma membrane is amphipathic. Similarly, here also amphitrichous. It can be single or a tuft of flagella. A tuft is nothing but a group, a bunch again. A single or a tuft of flagella present at both ends of bacterium. That is amphitrichous. Then we have lophotrichous. What is lophotrichous? Lopho is when a tuft of flagella or a bunch of flagella is present only at one end. That is lophotrichous. Now, if you see the example here, if you see the diagram here, a group or a bunch, okay, a tuft of flagella is present only on one end. Next, we have Peritrichous is when it's surrounded by flagella all over the body surface. The word peri itself says perimeter, right? Perimeter is what? The surface. So here also peri means on the surface. Throughout there is flagella that is present. So this is peritrichous, on the surface. I hope this classification is also clear. So from the components of a bacterial cell, we'll continue in the next video lecture because it will go into a little detail. So I hope you enjoyed the video and liked it. If you have, kindly subscribe and like the video, and also do share it with your friends and relatives. Thank you so much.